Okay, so we were talking about pH and biological systems and all of that. And so we had said neutral, pure spring water would be would be pH of 7. But now when we talk about biology and this kind of systems like us, what we find is that we are slightly alkaline. So what does that mean? That means we have slightly fewer hydrogen ions than pure spring water, and the pH for humans, for blood and body fluids, would be 7.35 to 7.45. Now, if you within any biological system, there's going to be a limit to what you can tolerate. So what you can tolerate is, there's a limit to that. And so but you can tolerate some variation. So when we talk about homeostasis, remember we had said homeostasis is fluctuation around a set point. So if we had talked about pH 7.35 or 7.45 being sort of the set point, then the fluctuations around it, if it's slightly acid, it'd be acidosis. If it's a little bit off of that, as a higher number, it would be alkalosis. And the way that you maintain pH like that is to have buffers, and basically there are mechanisms in place to absorb excess hydrogen ions, or to release hydrogen ions if you need to maintain the pH at the correct level. Now, we're not going to talk just a whole lot more about pH, but it's something that's important to understand. We are going to talk about types of reactions. Now, when we talk about biological systems, what we find is that in biological systems, you have what we call biomolecules, and biomolecules are big. They're really huge molecules, and they're generally made up of pieces. And what we would call these would be the monomers, the pieces that can be put together. So if you're building a big molecule made up of a whole lot of sugars, a polysaccharide is made up of a whole lot of individual sugars. If you're building that, it would be what we call synthesis. And if you're, and that would be like something in your body building something like glycogen, a storage molecule for sugars in your liver. You would be synthesizing, pulling all these individual sugars together in a long chain. Now, later on, if you needed sugar, you would be decomposing that to release the sugar into your bloodstream, and that would be a decomposition reaction. Then we have a third kind of reaction that happens in biology, where you have a multi-piece chemical here and one here, and they might trade apart. This one might take something off of it and give it to that one over there. And that would be what we call an exchange reaction. So these are the three types of reactions that we talk about. So when we talk about organic molecules, what we're really talking about is something we would call a polymer. So a polymer is made up of monomers. And I was just mentioning that a polysaccharide is a sugar made up of a lot of simple sugars combined together, synthesized together. So you can sort of generalize that, because we're not just made of sugar, we're made of protein, we're made of fat, we're made of nucleic acids as well. So carbohydrates, proteins, nucleic acids, and fats are the major categories lipids, fats equal lipids. These are the major categories of biological molecules. And so when we talk about this, we can talk about synthesis of a polymer. That would be you'd have each of them adding more and more of these monomer building blocks, or decomposition of a polymer, taking it apart. And interestingly, when you put molecules together, where you sugar molecules together, or proteins, or when we build any of these biological macromolecules, when we combine them together, every combination creates a single molecule of water. And so when we talk about synthesis reactions, we call it dehydration synthesis. And what it means is you take these two things together, and when you put them together and combine them, you produce a molecule of water. And then when it comes to breaking them apart, in order to break them apart, you have to put a hydrogen here and an OH here. It requires water, so it's a hydrolysis reaction if we talk about pulling it apart, because we have to have a hydrogen on one of them and an OH on the other of them. So we'll look at some details of that, because I'm sure that was not particularly clear. In today's lecture, we're going to continue on, and we're going to talk about carbohydrates, and we're going to talk about the genetic material nucleic acids. So we'll talk about carbohydrates next.